So thank you very much, everybody, for being here with us today. We are following the series of webinar Structure Meets Function that at this stage of the seminar, it is showing how users that have been visiting in a number of different modalities, the different centers in the different countries, have achieved very interesting results. And with that, we really expect to encourage you to contact Instruct and to make sure that your research can access top of the line infrastructure and equipment. So today we are going to have with us essentially three speakers. The three speakers have visited Instruct Spain and have obtained each of them results in different manners. I mean, either coming with the protein or with the images in different ways. I'm not going to repeat the names or the title of the talks. You are going to see one by one. And let me introduce a little bit what Instruct is. Instruct is the European infrastructure for integrative structural biology. And we provide access to top-level research infrastructure able to obtain an atomistic understanding of your macromolecule. I mean, X-rays, NMR, cryo-electron microscopy, biophysical characterization. We're going to see that this can happen in a variety of manners, and each of the speakers will have used our resources in a different manner. We are going to see how the power of internships is important, and how really is the best way to immerse yourself into the environment and to really get the deepest training we can offer. But we have courses, we have 1 month courses about 4 times per year, and also Instruct per se has a small research and development funding for seed money that is extremely competitive. I mean, we have a very high quality, and the last call easily got a success rate of 10%. So we are talking of very interesting funding for this, for starting new ideas. The Instruct Center in Spain is complex because it has 2 facilities. It started with the one you have on the right, the Instruct Image Processing Center, that really is about how do we extract the most of information from your images and assume that you have already obtained them somewhere else. And the latest addition that as facility was last year is the cryo-EM facility itself, is the ability to use a number of transmission electron microscopes from 200 kilovolts, 300 kilovolts, and we then obtain the images that then could be very easily processed at the image processing center. Really, we have a very healthy mixture of countries. Certainly our impact among Spanish scientists is important, but it's less than half. And we have, for the rest, we have visitors from all over the world. And I don't want to take any more time. It's just that I encourage you to register as an Instruct follower if you want, with an application that is the area, and from there on you will have news of every activity we do. So really I don't have much more to say, and really it would be the role of the speakers to show the power of Instruct. And our first speaker is Ryan Jones. He's going to talk about a very important virus with non-negligible pathologies if you happen to be infected in some countries in the world. So Ryan, please go ahead. I'm going to stop my sharing.
Thank you very much, Jose Mari. So I'm going to, share my screen. Okay. Okay, so can everyone see?
Perfect.
Okay,
We see
fantastic, and you can hear me? Great. Okay, so yeah, I'm very happy to be able to talk today because I've benefited enormously from the Instruct facilities at the CMB over the last few years. So today I'm going to talk to you about my work on characterization of the RNA capping pathway from chikungunya virus. So I'm from the Regera team in Marseille, and we're interested in studying viral replication. And one of the viruses that we study is chikungunya, which is a virus that is transmitted by members of the Aedes mosquito family to the human host. And it causes symptoms that include fever and very severe joint pain. And currently there are no existing antivirals against this virus, and it's quite an important health burden. So one of the best ways that we can kind of design new therapeutics will be in understanding the viral replication cycle. So this is a really simplified overview of the chikungunya replication cycle. And basically this virus has a single-stranded positive sense RNA genome that is released into the cell upon fusion with the cell. And this genome is capped at the 5' end and polyadenylated at the 3' end. And so in this way, it kind of mimics the cellular mRNA, and it can be translated by the host ribosomes. And in particular, this cap structure is important for translation and stability of the RNA. And the first reading frame within this genome encodes for non-structural proteins that are involved in replication. And these are cleaved by a viral protease within NSP2. And following this cleavage, they either go to separate compartments within the cell, and here they have different roles in infection, or they come together to form active replication complexes. And this happens exclusively at the plasma membrane in these little invaginations that you can see here. So these replication organelles form a protective microenvironment for the synthesis of the RNA. So if we look at the replication complex in a bit more detail, when I started this work years ago, then very little was known about the structure of these complexes, but we had a lot of information about the individual biochemical functions of the NSPs in replication. And NSP1 has two major roles in viral replication. The first being addition of this 5' cap structure, and it does this via a non-canonical pathway, which I'll go into in a bit more detail in a bit. And it's also the NSP, the only NSP that has affinity for membranes. So it was thought to be the membrane anchor for this replication complex. And there was a lot of evidence in the literature that membrane binding and RNA capping functionality might be coupled, but we didn't really understand why. And what we were able to show was that NSP1, when it's in contact with the membrane, it oligomerizes into ring structures where you have 12 copies of the protein in a single ring. And this oligomerization is crucial for the capping activity of the protein. And we solved the structure of these using single particle analysis and cryo-EM in collaboration with the people at the CMB a few years ago. So in particular, Rocio was very heavily involved in this work at the level of grid screening and kind of teaching us cryo-EM. And once we had the structures of these rings, we were able to use some cryo-tomography data of reconstructions from replication organelles of another virus, Flockhouse A virus, and show that NSP1, in addition to its capping functionality, is forming a pore within the membrane that gates the entrance to these replication organelles. So it controls the access in and out, and probably serves as a platform for assembly of replication complexes. So once we had the structure of these, we wanted to understand a bit more about the RNA capping pathway. And as I was saying, NSP1 does this in a non-canonical fashion compared to kind of cellular and a lot of viral enzymes. So typically within the cell, you have transfer of a GMP moiety from GTP to the 5' end of an RNA. And this is done by a dedicated guanyl transferase enzyme. And then this is subsequently methylated using S-adenosine methionine as a methyl donor at the N7 position and sometimes the 2'O of the ribose by different methyl transferases. In the case of NSP1 from alpha virus, it does everything. So it has methyl transferase and guanyl transferase activity, and it also kind of reverses the order of these reactions. So it begins by methylating the GTP at the N7 position. It then forms a covalent complex with the N7 GMP cap structure. And finally, it transfers this to the RNA. So you have methylation, guanylation, and then transfer. And so if we look at the structure of our NSP1, basically, the core of the protein is built around quite a conserved methyltransferase fault. And so this is highlighted in yellow and orange here. And you can see in the context of the rings, these capping domains form the crown above the ring, above the membrane. And I've overlaid it here with a capping domain from a canonical methyltransferase enzyme from E. cuniculi. And you can see in general, they overlay pretty well. And then NSP1 has evolved additional domains that confer other functions. So notably these blue helices that are involved in formation of the pore, and these red loops that are involved in finding spikes that go into the membrane. And you have 12 active sites around the ring. And each of these active sites is formed from a bilocal pocket, whereby you have a pocket for binding of S-adenosine methionine facing the exterior of the ring, and a pocket for the GTP facing the interior of the ring. So, as I said, NSP1 has both methyltransferase and guanyl transferase activities, but it looks more like a methyltransferase. So it really wasn't clear how it was transferring, how it would have these two functions. And additionally, in the literature, there was a lot of mutagenesis data that implicated this histidine residue that I've highlighted in red here as the catalytic residue that forms a covalent bond to the alpha phosphate of the M7-GMP. But if we overlaid NSP1 structurally with other methyltransferases, and then looked at where the GTP should be binding, it looked like this residue was far too far away for this to happen. So we wanted to know how this was happening. In addition, there are 12 of these active sites around the ring, so we wanted to know how many of them are active. So it was clear that for this, we need quite a lot of cryo-EM data. And because we don't have a high-end cryo-EM microscope currently at my institute, and we'd already collaborated with the people from CNB, I actually applied to have an Instruct internship to go to the CNB for a few months. So there were kind of two parts to my internship. The first was just sample preparation of grids. And for this, I purified my protein in my host institute, and then I gel filtered it on site, incubated with the substrate, and I learned to freeze my own grids with the VitraBot there. And this was done with the expert help of Noe and Javi from the facility. And these guys are really fantastic. And also my protein needs a support to go onto the grid. So I also learned while I was there to make carbon and graphene oxide grids. And so these are now techniques that I've been able to take back to my own institute and use. And then we collected a lot of data sets. So I collected 3 data sets from this internship, and then some other data sets from other visits. And again, this was done in collaboration with Rocio, the EM manager there. Yeah, normally we collect on a Talos, and if the data sets look promising, we then go to the ESRF to collect at the cryos. And then for the second part of my internship, I asked for some time using the I2PC image processing facilities. I did this because they have some software that I don't have in my institute. So I've mainly been working in Relion, but for example, I also wanted to try CryoSpark and Cryolo for part of picking. And the really nice thing about the facilities that they have here is that they have CPN that works really smoothly. And so you can really easily pass your data set between different programs. And for reasons that I'm not going to outline today here, I found that some programs are better for doing different steps. And the other advantage, of course, is that you can get really expert advice from the people there who really know what they're doing. So for example, Roberto helped us with some things a lot. So I'll talk about the structures. So for the first step of the reaction, where the SAM methylates the GTP, because this proceeds in the absence of cofactors, then we actually collect data sets with the substrates separately. And the SAM binding site, as I was saying, is the SAM binding pocket faces the exterior of the ring. And if we overlay the NSP1 structure with canonical methyltransferases, so again, this is the ECM1 protein in green, and this is the NSP1 in yellow. And you can see that there are several alterations in the site which effectively make this site more open to the solvent. So you have this movement of the helix, and there is some outward movement of some loops here. And the overall consequence of this is that it makes this site more open to the solvent. And so the local resolution in this region is really quite poor. It's over 3.5 or something. So it makes modeling into this site quite difficult. But anyway, and you can see the density is a bit scrappy, but anyway, we could see a SAM definitely bound, and we were able to model the purine ring and the ribose into the density, but then we kind of lose this density for the methionine. And actually many of these contacts that are being formed to the SAM are conserved in other methyltransferases. So notably this proline residue and this glycine that are part of these highly conserved SAM methyltransferase motifs. But it's interesting to note that most of these contacts are van der Waals contacts. The only possible hydrogen bond you have is to the ribose hydroxyl. And also this aspartate residue, which normally hydrogen bonds to the N6 of the SAM, is in our structure pointing away and coordinating a water molecule. So overall, it seems that the SAM is very flexibly bound here. We can't see anything for the methionine or residues that should be contacting the methionine, and that these contacts are quite weak when it's bound on its own. And so we wondered whether we could improve this with classification. Now, the problem with doing this with traditional 3D classification approaches is that unless you have the same number of monomers modified in the same sites and appreciable differences, these will be rotationally averaged even if you reconstruct it in C1 without symmetry. And so what I did was symmetry expansion, whereby you apply a matrix to position each of the protomers in the same position. And then I did density subtraction around a masked region, masking out the capping domain, and 3D classified with the particle poses fixed and whacking up this T value. And again, I got some advice from Roberto Malero for doing this. And what I see, I tried with different numbers of classes. And what I see is that basically you do see differences between monomers and different monomers within different rings. I have some classes where, like in our main structure reconstructed in C12, you have clear density for the purine and the ribose, but the rest of it, you can't really see anything. And then I have another class whereby you can see the whole of the SAM. But the interesting thing about this class is that the region around the SAM looks quite disordered. So it seems that the binding of the SAM is kind of destabilizing this site somehow. And indeed, the local resolution in this region is actually worse than in my APO structure. And then I have a third class whereby this pocket looks kind of empty. So yeah, overall, it looks like the SAM on its own is bound very flexibly. For the GTP, this site faces the interior of the pocket. So the density here is much better, the local resolution, it's better defined and it was easier to build. And if we compare structurally with other methyltransferases, so NSP1 is in yellow again here and ECM1 is in green, you can see that in NSP1, this GTP binding pocket is much deeper. And we actually see in our structure that the GTP is bound much further in, and you have this very greasy pocket where you have aromatic residues flanking it. And the overall consequence of this is that it brings this alpha phosphate residue closer to this catalytic histidine than it would be in a normal canonical structure. But equally as with this, equally as with the SAM, we see that while we can model in very easily the purine and the ribose, these phosphates look quite disordered. And again, most of the contacts are made in this region. And something that is interesting to note in this structure is that if you really whack up the contour levels to see the SAM binding site, we actually see some continuous density going from the GTP into the SAM binding site. And this is a bit weird because I didn't add any SAM. I don't usually see SAM co-purifying with my protein in the APO form. So I'm not sure what this is. But for me, this suggests that to have this GTP bound there, you need something kind of holding it in. Okay. So, and just to reinforce this, for the second step of the reaction, so this is where the M7 GTP forms a covalent complex with the NSP1. I recorded a data set with M7 GTP and the SAH, which is needed for this reaction. No one knows why. In the absence of magnesium, so they wouldn't react together. And here, although this is a Talos data set and the overall resolution is lower, the density corresponding to the substrates is much clearer. And you can see that this methyl group on the GTP is quite nicely positioned with the SAH sulfur, which would have been the donor. We also see, although a lot of these contacts are conserved with the SAM and GTP structures, there are some side chains that move. So notably this D138 residue that I was talking about in the SAM site, it now moves in to contact the N6. Equally, this glutamate hydrogen bonds to the methionine, and you have this arginine coming in to fix the phosphates. So it seems that, and we also see a slight rotation in the binding modes of these substrates. So it seems that when you have them together, everything is kind of clicking into place in the active site. And again, a consequence of this is to bring this alpha phosphate a bit closer to this catalytic histidine residue. It's still not quite close enough for nucleophilic attack. And I think for that, I probably need to record a data set with the magnesium. But this fits with biochemical data that I have in the lab and other people, which shows that the GTP on its own, or M7 GTP, can't be transferred to the protein in the absence of SAM or SAH. So the GTP transfer I can follow using radio-labeled GTP, and you can see that it's only really efficient in the presence of SAM. And this M7 GMP transfer I can follow with an anti-M7G antibody, an SDS page. And again, you can see you do have some transfer in the absence of SAH, but it's much more effective with the SAH. And before, no one really knew why. And I think it's because the SAH is important for positioning of this M7 GTP or the GTP in this site. And this could explain why the order of the reaction goes on in the way that it does, and you can't just have GTA's activity first. So then we also recorded a data set with the covalently reacted form. And in this case, sorry, I'm currently analyzing the data from the Cryos, but this is data from the Talos at the CMB. And we do see density connected to this histidine, but again, if you do symmetry expansion, this is not in all of the sites. In some sites, it looks as if you have M7 GMP bound, but it's not covalently bound to the histidines. This suggests that this bond is quite labile. And again, on a kind of continuation on this theme, we included SAH in this reaction because you need it, but although it's not reacting with the protein, it stays bound within the site. So again, this suggests that you kind of need these two linked together. So for the final step of the reaction, we wanted to look at the transfer of this cap structure to the viral RNA. And so for this, again, I use alpha phosphate GTP, and I can follow this transfer using SAM to the NSP1 and then to the RNA. And I use an RNA sequence that corresponds to the 5' end of the chikungunya genome. And then I can follow this using radioactivity and RNA gels. And so you can see here, this is capping of the 15-mer RNA, the sequence here, by the vaccinia capping enzyme, which we use as a positive control. And we also see that the chikungunya is able to cap this RNA, both in the diphosphate and triphosphate forms. So the triphosphate form is what we normally get from in vitro transcription, and the diphosphate RNA is supposedly the substrate for this reaction. But anyway, yeah, it caps both. And then I also tested capping of RNA from another alpha virus, Venezuelan equine encephalitis virus. So this is from a different clade. It's a new world virus and not an old world. And you can see again, the NSP1 caps this very nicely. However, I then took a completely unrelated viral sequence from Bunya virus. And sorry, I haven't got the whole sequence here, but it definitely begins GUG. And in this case, although this is capped by the vaccinia, we don't see any capping by NSP1. So this suggests that this transfer of the cap structure is sequence dependent, sequence specific. And then we also wanted to look at differences in the RNA length. So the chikungunya viral genome folds to form a highly conserved hairpin at the 5' end, right near the beginning. And this is predicted to be quite a stable structure. And you can see our 15-mer is cutting this hairpin in half. And whereas you have transfer quite efficiently to the 15-mer, you can see that this really plummets. You don't really see any transfer to the 27-mer. And unfortunately, I haven't been able to do the structure of this protein with the RNA. But for me, this makes sense based on the structures that we have. You have these active sites that are connected to the exterior of the ring via a positively charged channel, where we think the RNA is coming in to collect this covalently bound cap structure. And if I model an RNA into one of these cavities, you can see it looks like it fits quite nicely, this RNA sequence. But for me, it looks like this is too narrow to accommodate a double-stranded RNA. So this suggests that the RNA capping is happening co-transcriptionally as the RNA is being passed to the NSP1 and not following transcription of the whole RNA once you have the formation of these structures, sorry. And finally, you can model in about 3 to 4 nucleotides, and it's interesting to note that the 5' sequence of the RNA from all alpha viruses is really highly conserved in these first 4 nucleotides. And then we had a bit of an unexpected result, because during my capping reactions, I was seeing a lot of production of M7G-MP, which is not a product on the reaction pathway. And so I was wondering if this was coming off the enzyme, which based on our structures could be a possibility, or if this was coming off the RNA, and whether the reaction could be going in reverse. And so to test this, I capped the RNA with a vaccinia capping enzyme. I then precipitate it to remove all of the substrates. And then I incubate without and with NSP1, and also with a vaccinia decapping enzyme as a positive control. And you can see that with the 27mer, across all concentrations of NSP1, you don't see any loss of signal, and we see complete loss of signal for the DCP1. Sorry, I've cut this gel off on the end, but with the NSP1, with the 15mer, you can see that as you increase the concentration of NSP1, we seem to be losing some signal here. I have some gels which suggest that the RNAs are stable. So this could suggest that the RNAs might be being decapped, and maybe this reaction is reversible. So I'm currently running some more experiments to look into this. And so in this case, what we would have is the RNA is being capped co-transcriptionally, and then once you have the formation of this hairpin structure, it can no longer extend back into the site, and maybe this is a kind of protective mechanism for preventing the reaction from going backwards and driving it forwards. And of course, it would be interesting to test whether it's also capable of decapping cellular mRNAs, because most cellular mRNAs begin with this AUG sequence that you also have in the alpha viruses. And some viruses actually decap cellular RNAs to shut down translation. This is not something that I think has been documented for alpha viruses yet. And also, of course, it has interesting implications for the viral RNA itself. So I told you in the beginning that the chikungunya viral genome is capped at the 5' end. And this has been the dogma for years, but it's not actually strictly true. There have been some interesting studies recently that suggest that you have a mixture of populations of different viruses produced from cells, some that are highly infectious and where the RNAs are very stable, and some that are much less infectious and the RNAs that they contain are rapidly degraded upon delivery into the cell. And the main difference that they found between these is the proportion of capped RNAs within the viruses. And this paper suggests that as little as 15% of RNAs are capped in virions produced from mammalian cells, and 43% in virions from mosquito cells. So we wonder that whether NSP1 could be involved in this process, and whether it's simply a consequence of this reaction being reversible, or whether these RNAs have some kind of role. And what is interesting to note is when they increased the capping efficiency of NSP1 in the context of a virus, they saw that this was actually detrimental to infection. So this could suggest that these RNAs may have these uncapped RNAs may have a role in infection, but we really don't know what this is at the moment. And if anyone has any ideas, though. So in conclusion, NSP1 has kind of reconfigured its active binding pockets relative to normal methyltransferases in order to evolve methyltransferase and guanylyl activities. And based on the structures that I have, I think that binding of the substrates together is really important for the stabilization of the active site and correct positioning of both the substrates and the residues, and particularly for this positioning of the GTP and the alpha phosphate near the catalytic histidine. So I think this could explain why you need SAH for the second reaction. It's not allostery necessarily, as people thought. And it could be for this reason that the reactions proceed in the manner that they do, where you have methylation before guanylation. Finally, NSP1 RNA capping is sequence and structure specific, and it looks like it occurs co-transcriptionally. And it looks as if this capping reaction may be reversible. And so we wonder whether this could contribute to the formation of these uncapped RNAs that you see in some variants. And so, yeah, I'm not going to talk about the number of monomers modified per ring. We think that there are differences, but I'm not sure how this links to the RNA. So thank you for listening. And I'd like to thank my boss, Juan, at the AFMB, but really in particular, all of the people at the CMB who've been really fantastic throughout this project and internship. And we really wouldn't have got it off the ground without them. So Rocio for help with data collection, Javi and Noe, and Javi as well for data collection. And then Michael Hons has also been helping us with data collection at the ESRF. So thank you for your attention, and I'm happy to take any questions.
Thank you very much, Ryan. And we have several questions in the question and answer. Okay. Nick Papalikonau is asking for two things. One, if NSP1 or other non-structural proteins have non-structured regions, are known to have non-structured regions, and how do you think these non-structured regions appear in cryo-EM?
Yeah, so NSP1 does have a disordered region at the C-terminus. There are about 60 amino acids that I don't see in any of my structures on its own. And I'm convinced that this has a role in interacting with other NSPs. I think that when we add them in, then this will probably become ordered and we'll be able to see it. But for the moment, we really can't see it. And there's just no density there whatsoever. So you can't even look for it using focus classification or something.
Yeah, really, cryo-EM is not a technique to see non-order or non-structured regions, unfortunately. Then there is a question that you may know or I may complement, is how expensive is a cryo-EM microscope? Is a very specialized type of facility?
So yeah, they're super expensive. We don't have one at our institute, and that's why I'm enormously grateful to Instruct for funding this internship. And I think it's really important that you have these kind of initiatives so that people that don't have these high-end microscopes in their institute can access them. I don't actually know a price for cryo-EM, probably.
Well, it can be between 1.5 to 4 million euros, and then you need easily 300 to 400 euros, 1,000 euros per year for the maintenance contract. So they are expensive. Mm-hmm. Then Bram Koster is asking a question on procedures or workflow to work. You have used the Talos at the CMB, and then you are using now, I think, at the SRF, cryos. How do you find the interaction? I mean, how do you find that the fact that you have acquired knowledge and training at the CMB is helping you now with other microscopes that are not at the CMB but at the SRF? How does it work?
So yeah, I think it's still important that users process their own data and learn to do this, but these types of centers are really, really important for learning this. So for example, both the people at the CMB I've tested before with questions about data processing, and Michael at the ESRF. Because if you don't come from a cryo-EM background, then yeah, it's very important with your experience. So yeah, I mean, it is important. It's really important, these collaborations. I can't emphasize how important they are.
And I can say that actually we have almost every 2 months a teleconference with the SRF to be sure that the software is being used at the facility, CPM is being used at the most, and that we are receptive to whatever need it happen. Bram Koster, he congratulates you, as you see, for your achieved resolution. And well, it's now, if you have thought about tomography.
So we would love to do, yeah, we would love to do tomography, but So this is not something that we've started doing. We don't really have the resources yet, but there was a really nice paper that came out on alpha viruses a few weeks ago from Hua Chu's lab and Dahai Luo, where they've done tomography with these replication organelles from infected cells with chikungunya. And there they see our NSP1 rings, but they've also been able to dock some other NSPs from the replication complex. So yeah, I think tomography, particularly for looking at these kind of complexes that are embedded in the membranes, it's a really super technique, and it's something that we'd love to get into.
Yeah, and you may have interacted with Javi Conesa, and Javi is the expert in making lamellas and everything at the CMV. So you see that you keep more questions coming, but I'm sorry, I think it is the time to go to the next speaker. So thank you very much, Ryan, and if you wouldn't mind to answer in the question of question and answer part. Thank you very much
for the invitation.
Thank you. So the next speaker is Simone Weyand, and she's going to present another way of interaction with Instruct Spain, and we are going to see a very brilliant result. So please, Simone, the floor is yours.
Hang on.
We see a very nice rhinoceros.
Yes. Hang on. So, this one. Okay.
Yeah.
Okay. Can you
go to present to full screen mode if possible?
Yep.
Perfect.
Okay, super. So yes, first of all, thank you very much that you asked me to speak today, because I think the results are really, really exciting and spectacular. And I gave it a bit of a vague title, From Structure to Function and Back, because it's still very much a work in progress, and you will later on see why. But basically, we hunted down a crucial membrane protein by cryo-EM, which is very important or literally essential to take up drugs against the African sleeping sickness, a neglected tropical disease. So neglected tropical diseases, just to give you a little bit of an idea, is a diverse group of different infections like trypanosomes or malaria, about 20 different ones belong in that group. And they are very often or mostly present in Africa, Asia, and Americas, especially Central America. They can be caused by viruses, bacteria, protozoa, or parasitic worms. And the big problem is really, and I realized that too with my project, that working on them makes it incredibly difficult to get funding for supporting your research. Of course, you can try to prevent these diseases from happening. For instance, you have clean water, filtered water, or proper housing, etc. But it's not always possible. And so it's really important that there are either vaccines available or some sort of small molecule drugs for treatment. Now, the African trypanosomiasis, or African sleeping sickness, is an insect-borne parasitic infection in humans and animals. Here you see people in Africa being affected by the sleeping sickness, and it really makes you very unwell and is fatal eventually if it is untreated. And likewise, also trypanosomes cause African sleeping sickness in cattle and the big, or in hoofstock generally. And the big problem, which actually is very little regarded at the moment, is very little known, is that it is a huge agricultural loss if cattle are infected with it, because as you can see, usually these in the areas of the disease, people hold cattle for the meat production, but as you can see, the animals, they get exceedingly skinny, so there is literally no meat to get from them. They still live, but the economical, the agricultural impact is massive. Now, it is caused, this disease, by the tsetse fly, which you can see here, a huge, nasty fly, very unpleasant, and the bite is really painful, as I experienced myself when I went to Ghana to do field work. So trypanosomiasis is also present not only in Africa, but also in South America, and known then not as African sleeping sickness, but as Chagas disease. So this problem is spreading across the globe. Now, Trypanosoma brucei is the species you can see here in blue, and then we have the red blood cells. And this substrain is actually the one which causes the human sleeping sickness. And there are two stages of the sickness. The first stage is the blood stage, which is about 1 to 3 weeks after the bite of the tsetse fly. And after that, there's the nervous system stage. It goes in the nervous system in the brain, but that's usually months later. And the diagnosis is still by simply taking blood samples and or liquid samples from the body and isolating the parasite. So it's very difficult if it is in the later stage, or much more tricky to actually track down what the problem is with the person. And so far, the most used drugs, pretty much the two only drugs on the market, there are a few more, but these are the main drugs, are pentamidine and melarsoprol. And pentamidine and melarsoprol are rather huge. You can here see the structure. And in the case of melarsoprol, you see it's an arsenical. And the problem with that is that basically the drugs are so strong that either you die of the disease or pretty much of the treatment if it is the later stage and it has to be treated with the arsenical. So the first stage can be only treated with pentamidine. The second stage is mainly treated with melarsoprol. But you need a treatment because otherwise it leads to death. Now, suddenly there was more and more common that the drugs didn't work. And so obviously everybody immediately spotted there must be resistant strains. And what happened was the whole trypanosome genome is sequenced of the wild type. And so it was resequenced of resistant strains, and the genome sequence and protein sequences, particularly later, were compared, and a mutant was confirmed, a specific mutant of all the resistant strains compared to the wild type strain. And that was a membrane protein, which seems to be the culprit why, if there is a mutation, the drugs are not taken up anymore. And therefore we thought, well, the only thing really that can explain how this molecular mechanism works is if we decipher the structure and then therefore understand the function, and then go back again and design new drugs, or the pharma industry is designing new drugs to treat this disease. So in the wet lab, we cloned in my lab the membrane protein of choice. Then we have a little cleavage site, a TEF cleavage site, and then we have a GFP protein and a His tag. The His tag is purely to purify the protein, and the GFP is simply to track the protein in the cells, and to have always the option of chopping off the tag and have literally the wild type without any tag attached. We have a TEF site here. We did expression trials in insect cells, SF9 cells, and you can see here after 1 day, 2 days, 3 days, and about 4 days, how actually the membrane protein is increasingly expressed. And what we did was we harvested usually after no more than 72 hours, 3 days. And you can see here, for instance, this cell, how beautifully the membrane protein is sitting in the membrane where it's supposed to be and not inside the cell aggregated. We then set up and optimized the purification and a size exclusion chromatography, because we already expected it in oligomer. And what we wanted to have is the pure state of the active oligomer of this protein, and all the monomers which were dissociating we wanted to to remove. So here you can see the elution volume, a nice peak, and we cut out roughly here because we had really a lot of protein. And then again, we confirmed all the solubilization step, then the centrifugation after the solubilization, the flow through, the wash steps, the elution steps, and eventually what went on the grids from after the FSEC. So we have nice pure protein, and then in-house in Cambridge at the Department of Biochemistry and also at the Nanoscience Center in Cambridge, we did preparations. And what we wanted to have is, first of all, we wanted grids with the wild type, and then we wanted to just optimize those grids. And you can see here a micrograph of those grids that worked nicely, or after some optimization nicely. And then what we wanted to do is understand how these two drugs, pentamidine and milsaperidone, are actually binding to the wild type protein, so that we see how are they taken up, because there was a big speculation still in the field. There is a big speculation, does this protein actually really take up the drugs, or is it something else that takes up the drugs? But we know from functional assays, if the protein is knocked out, the trypanosomes, if they are incubated with the drugs, are surviving happily, and if we knock it back in again and do a rescue mutant, they die again. So it is definitely this protein. Now, we prepared grids. We use glow discharge quantifor gold grids with 2 microliter protein volume, about 1.75 mg per ml. We did screening at the Thales Arctica in Cambridge in-house in the biochemistry department, and we did data collections on the Titan cryos also in the biochemistry department and also at the nanoscience department. So the reason why we used 2 different Titans was simply that biochemistry didn't have the microscope at the time when we started screening and collecting the data, but later on it was just very convenient to go in the basement and collect data in-house. And I have to say, I'm really forever grateful to the Instruct team and to Jose Maria Carazo's group, because they did really all the trick of all the processing. So we tried to process the data, and there was just on each and every single level, there was so much problems and so many hurdles that eventually I decided to apply to Instruct and just kindly ask if the proposal goes through, if Marcos and Roberto could have a look at the data and give their opinion if it is possible to process or if it is not. The other problem was that meanwhile the lockdown happened, and my wet lab was not open anymore, and therefore that is what we were left with. So luckily and very fortunately, I got this proposal approved, and I sent Marcos and Roberto the data to Madrid. And I have to say, I have visited Madrid and the group before lockdown, before this project, for another project, which was just brilliant. But we did everything for this project remotely, and it worked just really well too, because everybody is just so transparent and so easy to get along with that there was no problem. So what Marcos did was using Scipion for the processing, and here you can see the flow chart. For movie alignment, he used Relion. For particle picking, for instance, he used Cryolo, then CryoSpark, and then Deep Enhancer eventually. And 2D classes of the 3 different data sets. First of the wild type, you can see here a side view, here a top view. So beautiful 2D classes, also with milsaperidone bound, with pentamidine bound. And then the 3D volumes, which here you can see the side view on top, and then the top views underneath, which is beautifully shown that we see the central pore of this protein as we expected, and then a homotetramer with a pore in each of the monomers, as we really hope to see. The FSC curves looked also really nice for that resolution. You can see for the wild type milsaperidone and pentamidine. And then there was the big question, is now the drug visible or not? And we thought it should be a good chance that we can see each of the drugs, because each of them is in the single digit nanomolar range of affinity. And indeed, as you can see here on the volume of the milazoprol already, you can see here this little moiety in the pore of each monomer, which is representing the milazoprol. And here again, in the side view, nicely, even on the unsharpened map, already nicely seen, even more beautiful on the sharpened map. And this is what I wanted to say with work in progress. We literally at this moment have all three models of the backbone of the protein built, and now currently are fitting the drugs in there, or try to see how we can fit them best in there. Again, the pentamidine density, also you can see here the little moieties in each of the pores. Again, then here circled in red. And this is the model of the wild type, the full model here, and also just the C alpha backbones is beautifully arranged, is really as it was supposed to look. So it was a nice confirmation that we actually did things the proper way. And the maps, I have to say, really look spectacular. You can see here just from the backbone how beautiful these alpha helices look like. Even the loops, they are just so well resolved and just so stunning. We couldn't believe that at the resolutions of 3.2 to 3.6 angstrom of the three maps, we can see such beautiful maps. And eventually I would like to thank a whole lot of people, but first and forever grateful to José María Cavazo and Roberto and Marcos. They are real superstars, and I really mean this, because they made things possible which a lot of people told me, oh, you can't process this data, and they could. They just understood really well this data in each and every single step. Then of course Dave Stewart and the whole Instruct team for making the whole process so smooth to apply, and having this brilliant facility that if you get stuck, then you can actually ask for help and you get the help, and everybody's so hands-on and friendly and supportive. It's amazing. Of course, Modestus, who is a PhD student, and Sandeep, who is a postdoc with Tom Blundell, who helped me a lot with all the building, because so Modestus did the majority of the building because they have the access of the programs, and are happy to share with me all of my wet lab members for the really, really long, year-long work to get it on the grids. And then also all the other people here who were real mentors and supporters in one or another way. Thank you very much.
Thank you, Simone, very much for your very exciting talk and for your very nice words. And now we have some question in the question and answer. I think you can also open that section. The first one is on detergents.
Yes, so yes, detergents were used at the we did a screen of detergents. I left out that step because I wanted to have the focus on the data processing. We did a detergent screen in the beginning, and basically we did an analytical FSEC with a whole range of detergents to see which one is the best, and also which one is still binding the ligand, which for us was the important bit in order to confirm that the binding pocket is intact, and so the protein hopefully functional. So we used DDM, and then different lipids. CHS was then the one of choice to solubilize and purify the protein.
And then, Simone, you have a more involved question from Patrick Beckelburg on really mixing different.
Yes.
You probably have heard a lot in the lab.
So what is the benefit of using Relion for alignment? And so why we basically, why do we have all these different programs in the flowchart? So this is the beauty of Scipien, and I have to just stress this because I really am a big fan. And no, it's not a paid advert. So I really say this because I mean this. So because what really happened is we tried different programs for each step. And this was the combination of programs, which did in combination gave us the best result. So, for instance, simply for the particle picker, and I mean, Marcus is the much better person to answer this because he figured out all these tricky steps one after another. But for instance, for particle picking, it was really Crayola and it was by far the best. But then for the alignment, it was Relion and it was really by far the best. So each step was tried with different software and carefully compared by Marcus and Roberto, which one is the best and which one is actually giving us the most satisfying result. And to understand each step and not just run automatically through them with one program, I think was really the key to success for this project to work, because I got told by so many people it's not possible to even pick the particles. And as you could see on the micrograph, you can see the particles, and Instruct made it possible. With a contrast comparable to the micrograph shown, would the cryospark blob?
Well, essentially, if the micrograph were good enough for just using one.
Yes. So how and when did you add the drugs to the protein for? Okay. Did you have drugs? Did you add the drugs to the protein for cryo? Yeah. So we had the drugs, so we purified the proteins drug-free, and then we concentrated it before the size exclusion chromatography, and that's when we added it and incubated it for a few hours, then put it over the size exclusion column. But the size exclusion column was simply to actually separate the tetramer, which is our functional unit, from all the monomers and dimers. And that was why we did then a size exclusion chromatography. Yes, it was done.
Simone, we have a live question from Ryan. Maybe Ryan wants to make it directly.
Yes, sorry, I couldn't type it in the chat. Going back to what you were saying about the using different software incipient, I was wondering, did you try different sharpening methods for your maps for looking for the ligands? Because this is something that I found made a big difference. Yes,
we did, and we still do at the moment. And it's amazing how, for instance, Enhancer is giving, such a beautiful density for the ligand, which is visible very well beforehand already, before it's sharpened, but so much better when it's sharpened. And you're absolutely right. I fully agree. It's very well worth trying different sharpening methods.
Okay, thank you.
For the cryolo picking, did you train a network or use one of their pre-generated models? I have to say, Marcus is better to answer that, because I don't know at the moment, but I can check with him and let you know later, or Marcus maybe can type.
Probably Marcus trained the network.
I guess so. Yes, I guess so, but I'm not quite sure. Maybe there would be. Oh no, that's other.
Okay, maybe just we go to the last question from Walter.
Yeah.
Walter Keller. In your expression,
which is green color all over the place at 93 hours, does it mean that the cells disintegrate? No. So what happened is that just so much protein was expressed that the membrane was simply saturated, and whatever was expressed couldn't fit anymore and was then inclusion bodies. So basically it was aggregated in the cells. But if you purify that, it's very dangerous, because you purify that, and because you use such high detergent concentration that you will purify, solubilize out of the membrane, and also the inclusion bodies. And later on, you have functional and non-functional protein. And you don't want this, because on the grid, you can't distinguish and you can't separate that anymore by the purification. So it's really crucial to pick them before they have inclusion bodies.
Okay. I think that you have answered all the questions, Simone. Thank you very much. Thank you. And we go to the last speaker, to Javier. Javier, please, you have the floor. And Ryan, you may now lower your hand, just not to have a leftover. Thank you. Perfect. So Javier, you.
Hello, everybody. It's a great pleasure to be here with you today. So first of all, I want to thank Sean for organizing this event, and to Jose Maria, Rocio Ranz, and all the team at the Instruct Centres Spain for the invitation. I think this wonderful opportunity. Also, I want to express my gratitude to Instruct in general, because have been supporting our research for several years. We have been partnering Instruct Centres to access to different techniques, X-ray crystallography, SACs, but especially today, I will talk a bit about how we involve Instruct in our cryo-EM work. So I will talk about the main research line in the lab. That is about the allosteric mechanism in bacterial glycogen biosynthesis. And I want to advance that is centered in the regulation and the regulatory properties of an enzyme called HCPase. We have been exploring this enzyme from different aspects, biochemically, biophysically, and but strongly centered in structural techniques to unveil the mechanism of allosteric regulation. So I organized the talk in these 4 sections. First, I will present how we work alongside Instruct, first introducing our group, that is Structural Glycobiology Lab, how Instruct have been a key player for our cryo-EM studies, and the experience that we have with the Instruct Centres Spain, CMB Madrid. Then I will pass to the structure meets function topic of the day, that we are going to discuss allosterism. So I will give some definitions and the history and relationship between the structure and regulation of enzymes in general. I will pass then to the study case of my talk, that is the glycogen regulation in bacteria, centered in ADP glucose paraphosphorylase called HCPase and its function. And I will advance how we advance the mechanistic model in our study model, that is the E. coli HCPase and its regulation through snapshots that we obtain first by X-ray crystallography and then by cryo-EM. So first of all, our group, the Structural Glycobiology Lab, is led by Marcelo Guerin. And we are an enthusiastic group of researchers that currently, you can see, we housing several wonderful PhD students. We are based in the Bilbao area in the Basque Country in Spain, and we belong to the Biocruces Institute, that is part of the Cruces Hospital. And you can see that we are surrounded by the very beautiful nature. So our group is supported by different agencies to develop our research, government Spain, local government, NIH, and we have some partnership with the University of Luxembourg. So we also work in a very broad collaboration network in Spain, Europe, and USA. I just want to mention that just recently, a member of our group that is called Beatriz Astoy, that resides in Biocruces too, she became independent, but is still a collaborator of the group. So our scope is structural enzymology and chemical biology of proteins involving the biosynthesis and degradation of glycans. We work with carbohydrate active enzymes, studying their catalytic mechanism and allosteric mechanisms from the structural point of view, mostly glycosyl transferases, glycosyl hydrolases, and sugar nucleotidyl transferases, etc. So we're interested in bacterial physiology and pathogenesis, and we are very, very involved in the study of the glycogen biosynthesis in bacteria, cell envelope metabolism and remodeling, good microbiome and marine microbiome. And we use a multidisciplinary approach that includes X-ray crystallography, cryo-EM in the center of our studies, but with also biochemistry, biophysics, and molecular biology. So previously, we included Instruct in our cryo-EM workflow for high-resolution data collection, because we actually can do the protein purification, biochemistry, and the biophysics in our home lab at the time before we can also do negative stain vitrification and screening, and we have these built capabilities in the lab. And through the application of two different Instruct Centres, we collect high resolution data, and we can, and then we later move that data to our home lab, and we can perform the single particle analysis, reconstruction, and modeling. That was until 2020, that we were based in SIGBioUNE, that had a electron microscopy platform. But in 2021, the group moved to the Biocruces Vizcaya, and that have lacks from that kind of platform. And here it was when the Instruct Center Spain at CMB actually start to be a fundamental player for our cryo-EM studies. And we hope that also in the near future, when the capabilities are built, we can also apply to this center for high resolution data collection. So we are, as I mentioned, fairly new users of the platform. So I want to give our experience of how is work with the people at from the cryo-EM facility at Madrid. We are actually located 400 kilometers away from Madrid. So there's two main pathways that we take depending on the characteristics of the sample and the last steps of the sample to be prepared. So we either send samples directly by shipping on ice to the facility, and the same or the next day when the samples arrive, they can perform negative stain and vitrification, and then we can see the subsequent days, depending on the availability of the microscope, the screening remotely. The other way that we work is part of the team moves to the CMB, so we can do some on-site handling of our samples. And actually, when that happens, we usually observe the screening and the data collection on site. Then we transfer the data back home, and our group of experts analyze the data. And this have been a wonderful experience for us, because actually this cryo-EM facility in Madrid saved the day for us, because we can keep pursuing our research in cryo-EM. So we are very grateful to the Instruct team led by Rocio Ranz and all the wonderful guys that are in the facility. So now I will start defining what we study at the fundamental level, that is allosterism. So allosterism have been nicknamed by Jacques Monod as the second secret of life, because actually allosterism is about the transmission of information. So the signal transduction pathways and allosteric transitions that couple distant sites in a protein or macromolecular complex. So allosterism comprises changes triggered in the structure upon ligand binding, reflect the level of energy landscape, conformations, variations in the residue interacting networks, dynamics, vibrational mode, assembly, and so on. In particular, enzyme allosterism is really fundamental phenomenon in life. allosteric enzymes control and coordinate the cell metabolism and allow the relationship of otherwise unrelated reactions. Signaling among active sites and regulatory sites can be either positive, enhancing the activity of the enzyme, or negative, inhibiting the enzyme. This allows the feedback regulation of different pathways and allows the building of hierarchies in the metabolic network, and ultimately allows the virtual compartmentalization of chemical reactions. This is just to mention that actually one of the first time that this phenomenon was observed in a protein was by Gertner Corey in the early 30s in the glycogen metabolism by the activation of the glycogen phosphorylase by AMP. So talking in particular about the coupling between these two virtual compartments, that is the energy generation and the glucose energy storage. In the overall energy generation metabolism, we have different pathways, glycolysis, respiration, or photosynthesis, that generate high amounts of energy available in the cell from either glucose or any other species. And in the case of higher cell energy status, we actually know that glucose is synthesized back, and part of that glucose from the energy surplus can be stored in polyglucose storage polymers called glycogen and starch. Glycogen that is found in bacteria and animals, and starch in plants. And to coordinate that metabolic network, it is important that these low energy or high energy signals relate both compartments. In particular, for the glycogen metabolism in bacteria, the classical pathway for glycogen synthesis starts with the synthesis of nucleotide sugar donor ADP glucose from ATP and glucose 1-phosphate, and then the glycogen synthase elongates alpha-1,4 glucan linkages, and branching enzymes act upon to generate a ramified structure. Then to recover that glucose in a later time, the branching enzyme can act, and a glycogen phosphorylase upon that to recover that glucose that is stored. So again, then we can think about glycogen as a main chain of alpha-1,4 linkages of glucose, with often ramifications in 1,6. This creates these glycogen granules that are a kind of a fractal molecule with many reactive ends that allows to deposit and recover glucose very quickly. And importantly, the whole pathway in the synthesis pathway is regulated by the enzyme ADP glucose pyrophosphorylase. So this enzyme is a very classical enzyme, was firstly isolated in the 60s in the Leloir's lab, are tetrameric enzymes, and catalyze the nucleotide sugar donor synthesis. That is the first committed step for bacterial glycogen and plant starch biosynthesis. The reaction is rather simple. It's an exchange of phosphorus between ATP and glucose 1-phosphate mediated by magnesium that leads to the generation of ADP glucose releasing pyrophosphate. We know from structural studies and biochemical studies that the catalytic mechanism is triggered by the presence of lysines at the active site. So to study this fundamental mechanism, we use, as I mentioned before, equal IHPAs as a study case, that we know that the activator is a fructose biphosphate that signals higher energy content from glycolysis, and is inhibited by AMP that signals low energy from several processes in the cell. We know that this enzyme is an homotetramer, but at the time that we started these studies, that was about 8 years ago, we didn't know what it was the structure of equal GPAs, or where these regulatory sites were located, or which was the regulatory mechanism. So we started our studies with a crystallographic approach. We saw both crystal structures, and as you can see, this enzyme was very difficult to crystallize. And as you can see, the crystal structure actually result in very few, we have very few structures, and all of them were in a pitch-1 space group. We could see in the crystal structure variable amounts of these regulators for the inhibitor and for the activator. But first, I want to just disclose is that the overall organization of this enzyme comprises an N-terminal domain that is a GTA-like domain comprising the active site, and with a C-terminal regulatory domain. Through this C-terminal regulatory domain, protomers assembles into dimers, and finally, dimers assemble into tetramers. So it's a dimer of dimer tetramer. By X-ray crystallography, as I mentioned, we could locate the presence of the regulators for AMP and for SPP in a common allosteric cleft. In the case of AMP, we found that AMP is deeply buried in the cleft, surrounded by several residues, key residues that were known to participate in the regulation of the enzyme. But in contrast, we found that FVP was located more solvent exposed, but also participating in the interaction with some key residues. In particular, I want to mention that the importance of lysine 39 for the allosteric activation, and the arginine 130 from the neighbor protomer that we later verified that was a key element for the inhibition mediated by AMP. So with crystallography, we could unveil the regulator's binding site in this common regulatory cleft, but also by observing the location of the active site and regulatory site, we could identify some elements that participate in the allosteric communication. First, by active site to regulatory site in the same protomer pathways of communication, and also through elements that are related to the crosstalk between protomers. In particular, as an element that was described to be fundamental in the allosteric regulation, that is called the regulatory binding loop 2, this long loop that is here, we found it stabilized by a crystal contact, but it is known to be very flexible because that loop only can be modeled when participates in a crystal contact. Otherwise, we cannot see it in a crystal structure. So, doing these studies by crystallography, we could unveil the putative elements for the allosteric communication that we can say that are sensory motifs that here is in red, and the regulatory binding loops that decorates the active site here in cyan. And the crystallography picture gives us this type of model. regulators binding with partial overlap that supposedly should trigger some conformations or alterations of the active site, but that remains obscure for us because, as I mentioned before, both complexes crystallize in the same space group and have the same crystalline constraints. So then one question, I would say the main question that remains unsolved was which was the regulatory mechanism. So to explore the regulatory mechanism in a native environment in solution, we perform cryo-EM. So we solve both complexes by cryo-EM in native conditions. And as you can see, the maps look very beautiful. And we could locate the presence of the regulators in the cleft, that I had described before. So this is another model where you can see actually what is the relationship between the cleft and the active site. And already you can observe what is the conformation of this regulatory loop in the presence of AMP and also in the presence of FPP. So you can suspect how this communication between the regulatory cleft and the active site is performed. So, but when we compare the cryo-EM structures with the X-ray crystallography structure, we can see, for example, in the case of AMP, the complexes with AMP, that both the cryo-EM and the X-ray crystallography model actually give us the very, very similar answer. So the crystallography model was quite right. But in the case of the complexes with FBP, we saw that there was a change in the position of the fructose phosphate, and in the case of a cryo-EM structure, that actually appear located in the same position that AMP. And this has a rationale, because actually in our crystallization media, there was sulfate, and sulfate in higher amounts. So that sulfate remained bound in the position of the alpha phosphate for AMP, and it couldn't be displaced by the positive regulator. So this is a clear example of how cryo-EM map obtained in solution and native conditions reveal the physiological activator binding site, otherwise obscured by the crystallographic conditions. And more, actually we find more, that is that in the case of the E. coli HCPAs in complex with the negative regulator, the loops that participate in participating in the binding of the substrate forms a lock conformation interacting with each other. And in this way, not permitting the interaction of this loop that acts like a lead on the active site with the nucleotide, with ATP. But in the case of FBP, we saw that those loops are released, and actually we can, in the high symmetry model, high symmetry reconstruction, we could even trace part of that location of that loop. And in this is a case where cryo-EM in solution, actually exploring different type of reconstruction, allow us to reveal part of the flexibility of this loop at the active site that have been not observed before due to crystal packing. I'm not going to get into this detail, but also because we are working with this enzyme that now is in a native environment in solution, we perform exercises trying to reveal the changes in the residue interacting network. And in this way, we could track the pathways where the signal is communicated between the regulator binding site and the active site. So with the help of cryo-EM, we now could update this model for the allosterism of this enzyme, where in the case of the activator, the activator bounds and permits that the active site is in a conformation that is competent for catalysis, while in the case of the inhibitor, the complex with AMP, the active site remains in information with loops that are compromised in a lock mechanism that do not allow the catalysis. So, and these are just a few remarks about the talk. So, first of all, just to mention again that the Instruct have been key for the success of our cryo-EM research for higher resolution, but also the Instruct Centres Spain have been a very, very important player for continuing exploring the allosterism of these enzymes and others. For this seminar, the structure meets function, that I think we, through these studies, we have addressed a model to understand these fundamental phenomena, and obviously the advancing of this the unveiling this type of mechanism is we had to take the integrated approach of structural biology, where all techniques are important for disclosing this type of mechanisms. So, and that's all. Thank you for your attention.
Thank you, Javier, for your talk. And I see that there are no questions in the question and answers. I don't know if there is any presenter that would like to make a live question. And I would like to make you a question, Javier, is you have been showing many structural models and you have been relying on these structural models. Have you approached the topic of the map to model validation, and have you used actually tools, our FSEQ, the Q score of WATU, ways to know which part of the map would be more complicated for the modeling or series in Phoenix?
Yeah, we started this work before. We use validation tools, first exploring the resolution of the map in different regions with local resolution. We have been working lately with different sharpening tools as DPM Hanser to really see where are the variability of this model, and of course all the tools that are available for the validation that are included in several packages for refining structures. Also, we found very, very useful lately the use of ISOL, so we can actually validate the conformations in regions that have low resolution in the model. But in the case, in this particular case, this was very detailed work, mostly trying to address the model, build the model in different point groups, actually we can see some variability depending on how we build the model. For example, if it's in C1, C2 or D2, and depending on what we want to address, for example, I would say in the case of the activated state, we can see barely the tracing of the loops that are flexible. So we have let's say a lobe where we can model that loop. So probably the region where the loop is visiting more often the conformation. But to see the occupancy of the regulatory sites, for example, we also include an analysis in different point groups just to see where which cavities, which clefts were occupied by the regulators. In the case of AMP, that is in the inhibitor, we can see the density very strongly in all the regulatory clefts. But in the case of FBP, that is the activator, it looks like there's more density in the C1 map in certain locations. Actually, in 2 locations, it's very clear. And in the other 2 locations, it's a bit blurrier. But, you know, with the help of symmetry, we can enhance.
Yes,
also.
absolutely. You have that help.
So,
Yes.
but yeah, we perform all that type of analysis because also applying the full symmetry, there can be artifacts in the axis of symmetry, as you know.
Yeah. We have a live question from Ryan.
Yes, really nice talk. They're really beautiful structures. I have a biochemical question. I wondered if you've measured affinities of the ligands and whether this changes with the activator.
Very good question. So this is the thing. So the enzyme has a very high affinity. We have measured by ITC the affinity for AMP. And also we have performed thermal unfolding by CD in the presence of AMP, and we see that the enzyme is greatly stabilized by the inhibitor. In the case of FBP, it looks like the affinity is not so high. Actually, that's also observed by enzymology because it is required in the order of the 2 millimolars for the enzyme to being activated in the presence of AMP. So the ITC curves are a bit more difficult to interpret, but we know that it binds. So this is also an important thing to say, that we are working in native conditions, but we are actually working with an excess of the regulators to find it in place.
Okay.
I don't know if that addressed.
Yeah, thank you very much.
Thank you very much, Javier. Is there any other question, live or through question and answers? If not, probably we have reached the time in which people start getting tired and things like that, and we need a break. So we are going to close this webinar, and thanking certainly the speakers, Simone, Ryan, Javier. Thank you very much. Thanking Instruct for organizing the whole series and this seminar in particular, and thanking all of you that have been following, varying between, I believe, 80, 90, and 50. Thank you for all the time and for all the attention, and for. I can only hope that this will be another push to introduce Instruct as part of your research and produce research infrastructures in general as part of your research. They are essential, and they are there exactly for you and for improving your throughput and for improving the quality of the scientific result you are obtaining. Thank you very much. Bye.